Genetic drift changes allele frequencies by a random process. If we look at allele frequencies in 5 small isolated island populations, we can see genetic drift at work. Let's focus on a locus with 2 alleles, A1 and A2, with A1 occurring at a frequency of 1/2 when the islands were initially populated from the mainland. Random fluctuations cause the allele frequencies to change from generation to generation in a random manner that will be different from island to island. Even in the absence of natural selection, the isolated populations will diverge over time. Why are there random fluctuations in allele frequency? We can simulate the allele frequencies in a model very similar to the Hardy-Weinberg model. However, one of the 5 conditions of the Hardy-Weinberg model is not met. Population size is limited instead of infinite. As in the Hardy-Weinberg model, the parent population contributes alleles to a gamete pool, determining its allele frequencies. Then, gamete pairs are picked from the pool at random, determining the genotypes of the offspring. With the limited number of gametes chosen randomly, the allele frequency in a small offspring population may differ substantially from the allele frequency in the gamete pool. The larger the offspring population, the more random drawings from the allele pool will be done, reducing the importance of the random choices. Thus, we would expect that drift acts more powerfully in small populations than in large populations. 2 separate simulations with populations of size 10 and 1,000 individuals confirm this idea. Again, both populations start with 2 alleles, A1 and A2, each at a frequency of 50%. The simulation shows that allele frequencies change over time. The larger the population, the smaller the fluctuations in allele frequencies. This is similar to flipping a coin. The more often you flip a coin, the closer the observed frequency of heads will be to 50%. This explains why genetic drift acts more powerfully in smaller populations. As a result of genetic drift, one particular allele may reach a frequency of 100% in a given population. When this happens, we say that the allele has been fixed or reached fixation in the population. Genetic drift is a random process. Therefore, while it is certain that some allele will eventually be fixed in each population in this model, it is not certain which allele will become fixed in which population. In some of the populations, the A1 allele becomes fixed. In others, it is lost and the A2 allele becomes fixed. In this simulation, we reached fixation in all cases. If we look at populations of size 1,000, none of the alleles were lost in the first 100 generations. However, if we were to run the experiment longer and longer, in the absence of mutation and migration, each and every finite-sized population, no matter how large, would eventually become fixed for one or the other of the 2 alleles. To summarize, there are 3 general consequences of genetic drift. 1. In a finite population, allele frequencies fluctuate over time, even in the absence of natural selection. 2. Over time, some alleles are fixed and others are lost. 3. Separate populations diverge in their allele frequencies and in terms of which alleles are present.